Last lecture we talked about the structure of sugars, and we drew the Fisher projections and the ring projections of monosaccharides. Those are single sugar molecules. And then we also talked about when two sugars connect and make disaccharides via a glycosidic bond. This lecture will be building on that and discussing polysaccharides and glycoconjugates. Okay, polysaccharides are when many sugar molecules connect via glycosidic bonds. Glycoconjugates are when sugars connect to proteins. On the title slide, there's shown a couple of the polysaccharides that we'll be talking about today. Okay. On the right, we have the structure that glycogen follows. Also, amylopectin in starch follows the same sort of pattern of bonding. On the left is the structure that cellulose follows. Okay, and both of these polysaccharides are include only glucose molecules in them. So it's only one sugar that makes up these polysaccharides. But you can see that they're both quite different in their structure based on the glycosidic linkages that are involved, and their properties are also quite different. Right. So today we're going to be covering material from the second and third sections of the chapter, primarily focused on 7.2 polysaccharides, but we will cover 7.3 glycoconjugates. There'll just be a little bit less material from that section. Okay. But first, before we do that, we're going to start off with a couple of review questions, three review questions. Okay, so the first review question, draw the Fisher projections of glucose, ribose, and fructose. So to answer this question, you first need to know whether your sugar is an aldose or a ketose, and you also need to know how many carbons are in each sugar. So if we start with glucose, glucose is an aldose. So that means we're going to have to draw our aldehyde group at carbon 1. And you always start with carbon 1 at the top. Okay, and we'll draw an aldehyde here. Okay, and I'll just put a 1 there to stand for carbon 1. And now we have carbon 2, 3, 4, 5, and 6. Okay, so there are our carbons. Now, to answer the rest of this question, you need to know what side of the molecule you need to put your OH groups on. And to be really specific, you'd have to know, is this D-glucose or L-glucose? If you're not given that, you can really just assume it's the D form, because primarily all sugars in the body will be in the D form. There are some in the L form that are important, but the vast majority are in the D form. So if you're not told, just assume it's the D form. Okay, and glucose follows the pattern of right, left, right, right. So starting from carbon 2, the OH group would be towards the right, then towards the left, right, right, and right. Okay. All right. At carbon 6, we can just draw this carbon 6 as CH2OH, or you can draw it out explicitly. Hydrogen, hydrogen, and OH group. Okay, it doesn't matter what side you show the OH group there, because that bond can rotate, and all of those positions are equivalent. Likewise, the first bond up there can rotate, so showing the double bond on either side is fine. Okay. The only other thing we need to do here is draw in our hydrogens. Okay, and that would be the Fischer projection, although slightly scribbled, of glucose. Okay, now if we want to try to draw the Fischer projection of ribose, we have to remember what ribose is. Ribose is a 5-carbon sugar, and it is also an aldose. So we're going to start it in the same way as glucose, with an aldehyde at carbon 1. So that's carbon 1, this would be carbon 2, carbon 3, 4, and 5. At carbon 5, we can just put CH2OH, because that's our last carbon in the chain. It's not going to be a stereo center. Then you have to remember the pattern for ribose. And ribose is actually a very easy one. So the D version of ribose, all the OH groups are on the right side. So we can draw these all in the right side and put hydrogens on the left side. And that would be ribose. Okay, and finally fructose. Fructose is a little bit different because fructose is a ketose. So there's a ketone, the carbonyl is a ketone functionality. It actually happens at carbon 2, not carbon 1. So for fructose, we start with CH2OH at carbon 1. Then it's connected to a ketone group. And then followed by, so that's carbon 2 where the ketone is, then followed by carbon 3, 4, 5, and 6. Sorry, sometimes when I'm drawing it makes the page go forward or back. Okay, so at carbon 6, again we can just do CH2OH there. And now the pattern is only important at carbons 3, 4, and 5. And remember that fructose is just the ketose form of glucose. So it's going to follow the same pattern of, which side the OH group is on in the Fischer projection. So at carbon 3, 4, and 5 in glucose, 3, 4, and 5, we have the pattern of left, right, right. So that's what we would follow here. Left, right, right. And then we can fill in our hydrogens on the opposite sides. Okay, so that would be the Fischer projection of D-fructose. Second review question, draw the ring structures in the alpha anomers for D-glucose and D-fructose. Okay, so how do you answer this? Well, I think it really helps to draw the Fischer projection first. Okay, so we draw the Fischer projection. Okay, then from the Fischer projection for glucose, you make a connection using the OH group at carbon 5. So that would be the OH group. And it's going to connect with where the carbonyl carbon was. So in this case, it would be carbon 1. Okay. And so that makes a ring shown here in blue. Okay, the ring, the alpha anomeric carbon, would be at carbon 1. So that's right here. Okay, and that OH group can be either above or below the plane of the ring, right? And that's what the alpha and beta anomers mean. Okay. The other OH groups, where they are in relative positions. So you always draw the ring forms in terms of the convention is to draw that 6 carbon, so this is carbon 6, that CH2OH of carbon 6 is above the ring, just for a reference point. And I should probably label these other carbons. So that would be carbon 1, 2, 3, 4, and 5. Okay. So if this is carbon 6 and it's above the ring, what does that mean for the OH groups at carbon 2, 3, and 4? Okay, so if we look at carbon 2, that OH ring is pointing in here, so that's going to be below the plane of the ring. Carbon 3, the OH group is pointing out of the ring, so that would be above the ring. And again, carbon 4, OH is pointing down, or in, excuse me, so that's down. Okay, so the pattern we see here is OH group at carbon 2 is down, OH group at carbon 3 is up, and OH group at carbon 4 is down. So you can memorize this structure for glucose, or you can remember the process of drawing a ring form from the Fisher projections, which I think is a little bit easier because it kind of tells you the pattern there if you follow along. Okay, let's try fructose. Fructose is a little bit different because it is a ketose. So let me try to draw the Fisher projection without screwing up. So CH2OH at carbon 1. Carbon 2 is the ketone. And we have carbon 3, 4, 5, and 6. And carbon 6 is CH2OH. The pattern, again, is left, right, right. And I'm probably just, for simplicity's sake, I'm probably just going to draw the OH groups. Okay, sorry. Having trouble drawing that OH group without putting my hand on the screen. It's OH. Right, right. Okay. So I'm not going to draw in the hydrogens just because that can get a little in the way here, but this OH group is the one that's going to attach, and it attaches again to the carbonyl. You can think of it as the electrons in that OH group can do a nucleophilic attack on the carbonyl, which is electrophilic. Okay, and that forms a ring structure. So that ring structure is going to involve that bond, oxygen bond to that carbon, and then 2 more carbon-carbon bonds there. Okay, so you have a total of 1, 2, 3, 4. Okay, so now let's look at fructose. So fructose, again, it's a little different because it's a ketose. So let me try to draw the Fisher projection of fructose. And 6. Sorry, I always have trouble drawing down here at this part of the screen because my hand is on the screen. So C6 would be CH2OH. Okay, and the pattern from carbon 3 on would be left, right, right. And so from carbon 5, we're going to get an attack. You can think of this as a nucleophilic attack by the lone pair electrons on that oxygen. Okay, and that's going to make a ring structure that includes carbon 2, carbon 3, carbon 4, carbon 5, and the oxygen. So it's going to be a 5-membered ring for this structure. Okay. So again, this down here is carbon 6. We can use that as our sort of our reference point. Okay, 6 would be CH2OH, and above the ring, okay, it goes to carbon 5, which would be there. Carbon 5 goes to an oxygen. That oxygen connects with carbon 1. Then we have carbon 2, carbon 3, back to, goes to carbon 2. That's carbon 2. That'd be carbon 3, carbon 4, and carbon 5. Okay. Okay, now we need to tell, okay, what anomer is this? It's the alpha anomer. So what does that mean? It means that the OH group at the anomeric carbon is in trans form with the CH2OH group at carbon 6. So the OH group is going to be pointing down in this alpha anomer, and our carbon 1 CH2OH group will be pointing up. Okay. Now we need to look at the other positions. So carbon 3, the OH group is pointing out of the ring. So that means it's going to be facing up. OH is up. And at carbon 4, OH is going to be facing down because it's facing inside the ring in the Fisher projection. Okay. And again, I'm not showing all the hydrogens, so just because it'll make the structure a little bit more cluttered. Okay, so that would be the alpha form of D-fructose. And specifically for fructose, if you're drawing the ring form, you need to know if it's the 5-membered ring form, or which would be a furanose, called a furanose, or the 6-membered ring form, which would be pyranose. If you're not told, you can assume fructose is in the 5-membered ring because that's more stable, but it technically can form a 6-membered ring. In that case, the OH group at the last carbon would be attacking the carbonyl. Okay, moving on to the final review question. Draw the structure of the following disaccharides. Okay, so the first one is sucrose, and we're told sucrose is a combination of glucose and fructose. We're told that the linkage between the two is between the alpha anomer of glucose at carbon 1 and the beta anomer of fructose at carbon 2. Okay, so alpha 1 glucose and beta 2 fructose. So this one becomes a little bit more complicated because it is 2 anomeric carbons that are making the disulfide bond. The glycosidic bond. Okay. So if we look back at the ring structures we drew, okay, we want to connect them at the anomeric carbon, so carbon 1 in glucose and carbon 2 in fructose. Right, so we can draw a line between the OH group of one and the OH group of another, and that, in a sense, would be like what the structure is going to be like for the disaccharide if we were to draw that. So in this case, we could draw a line between that oxygen and that one, right? Then to show it, we would have to actually move fructose closer over here and rotate it around 180 degrees. And you would get something that looks, a bond that from oxygen that looks like this. That's kind of what that is shown like. But in this case, this is the alpha anomer of fructose. And we're asked to draw a connection between glucose and the beta anomer. So let me just erase those real quick. So we can draw this in the beta form. And this is going to look a little weird in red, but so the beta form, OH will be on the same side as CH2OH at carbon 6. Okay, so that would be the beta form. So now we're making a glycosidic bond between this OH group and this OH group. Okay, so that's where our bond forms. So now we need to rotate this 180 degrees when we draw our disaccharide structure. So what does that mean? Well, that means our CH2OH group, when we rotate that, it's pointing down now, but it's going to be pointing up. And this CH2OH group that was pointing up when we rotated 180 degrees will be pointing down on this far side of the disaccharide. Okay, so let's try to draw this, and I'll change the color to black. Okay, so first let's draw a glucose. Okay, a glucose, again, starting at carbon 6 would be CH2OH above the ring. C5 is going to be connected to an oxygen that goes to carbon 1, and then carbon 1 goes to carbon 2, 3, 4, and 5. Okay. That's our basic structure there. This is the alpha form of glucose, so our OH group is going to be pointing down. And this is our glycosidic bond, so I'm just going to draw an O there. Okay, the pattern, again, on glucose will be down, up, down at carbons 2, 3, and 4. Okay, so that's our glucose part, not showing any of the hydrogens, just for simplicity. Okay, then we draw our fructose. Okay, fructose is tough. Again, that anomeric carbon is where we're attaching to. Okay, so when we do that, in this picture, OH is up, but we want this to be, and this is anomeric carbon is on the right side. When we rotate it, the anomeric carbon will be on the left side, and O will be facing down. So that'll look something like this. Okay, that's carbon 1 in fructose, 2. This is carbon 1 in glucose. Okay. We'll have a CH2OH from carbon 1 pointing up. Then we can draw our ring structure. But these carbons now are sort of reversed from what the numbers that you would typically see. So this would be carbon 3, carbon 4, 5. Okay. When we rotate that, carbon 6 again is going to be facing down. So our CH2OH group is going to be facing down. What about the OH groups? Well, they're going to follow a same pattern. It's going to look the same, but remember the carbons are backwards, right? So now it's down at carbon 3 and up at carbon 4, where in the normal way we draw it with the anomeric carbon on the right, it's up at carbon 3 and down at carbon 4, but it looks the same here. But remember, those are different carbons now because it's drawn backwards. Okay, so this would be our picture of sucrose. Okay. Okay, let's try a little bit easier one, maltose. Maltose is where we have 2 glucose molecules attached via an alpha 1 to 4 glycosidic bond. And alpha 1,4 linkage is something we'll see again in this class today when we talk about polysaccharides. So our first glucose will look like this. And if I could draw this without touching the screen, that would be ideal. Okay, again, we have our down, up, down pattern. Okay, and this is an alpha 1. So that's going to be pointing down in the glycosidic bond. And then it's attaching to carbon 4 on the next glucose. So if we just draw another glucose molecule, Okay, we have again down, up, down. So this would typically be pointing down. So this is the oxygen that has to bond basically here, right? So we have to, they're both facing down luckily, so this is going to be easier to show. We just draw that bond down, okay? Connecting the two. Now at carbon 1 in the second glucose molecule here, this is a reducing sugar. So this ring can open up and you can have either the OH group above or below the plane of the ring. It can be in the alpha or the beta anomer here. And sometimes you'll see this depicted if there's a place that can have either alpha or beta at it. You'll see it depicted by like a wiggly squiggle line that just goes straight out to the OH, and that signifies it can be either alpha or beta. Okay. So this would be our picture for maltose. Okay, on an exam, I'm unlikely to have you draw these out by hand. More likely, you'll just see a picture of a disaccharide like this, and you'll have to tell me, okay, what is this, the bonding in it? You know, what are the monomers? What's the glycosidic bond? Or maybe I would give you pictures of multiple disaccharides, and you'd have to, and a list of these names, and you'd have to match the name to the correct structure. So that is the review questions we have. For this class being an online class, I suppose I could have you draw those on the final and then have you, take a picture of them and email them in. So you could still potentially have to draw these, but probably more likely is that I'll give you a picture of two, like a disaccharide, let's say, and I'll ask you to write out the name of that disaccharide. And that's when you need to be able to recognize between those sugars that you should remember, like glucose, fructose, ribose, and then how they're connected. So that's probably more likely a question that you would see than to actually have to draw them yourself. But being able to draw them, I think, really helps you understand and be able to see those structures when you just see the picture of them. Okay, moving on. Okay, polysaccharides, natural carbohydrates found in biology tend to be found as polymers. Okay, not as single sugars floating around so much as they are connected in a chain. Okay. They can be made of the same sugar, which we would call homopolysaccharides, or they can be made up of different different sugars which we would call heteropolysaccharides. Okay. They can be linear, and what we mean by that is, the same glycosidic bond repeating itself over and over, or they can be branched where you can have, multiple types of glycosidic bonds in a chain. Okay. They don't have a defined molecular weight. And that's important. These sugars, unlike proteins have a specific sequence which leads to a specific molecular weight. There's no template for polysaccharides. They can have a very drastic range of molecular weights. Okay. So this is just an example from the book, right? This on the left here, these sugars are all yellow, which means they're all the same type of sugar. Okay, so these would be homopolysaccharides. Glycogen and starch would be examples of homopolysaccharides. Okay, they can be unbranched, where they again have the same glycosidic bond repeating over and over, or they can be branched, And that's where you would have one glycosidic bond, and then at another point you have a different type of glycosidic bond happening that makes a second chain. Okay. So those are the examples of unbranched and branched with a homopolysaccharide. You can also have this with heteropolysaccharides where you have different sugars, 2, maybe even 3 sugars that are in the polysaccharide. Okay. The first polysaccharide that we'll talk about is glycogen. Okay, glycogen is what your body uses to store glucose molecules. Okay, so this is the storage polysaccharide in animals. And all glycogen is a branched homopolysaccharide of glucose. Okay, so there's only glucose in glycogen. Okay, these glucose monomers form alpha 1 to 4 linked chains. Okay, and then there are branch points that include the alpha 1 to 6 linkage. And this occurs about every 8 to 12 residues. Okay. The molecular weight of a glycogen molecule can reach several million grams per mole. Okay, and so here we see the structure. Okay, we have alpha 1 to 4 linkage within the chain of glycogen, right? So here's our alpha carbon, the alpha anomer, that OH group's down. Okay, there's an OH group here at 4 also pointing down, so this is alpha 1 to 4. Okay, the branch points are alpha 1, here's our alpha 1, and 6. So remember 6 is up here. Okay, so it's branching off from the OH on carbon 6 to another anomeric carbon 1 on a different glucose molecule. Okay, so that creates a branch. This chain, alpha 1 to 4 chain, continues on, okay, but it just branches off here. Okay, and from here you can get another alpha 1 to 4 linkage here. Okay. The storage polysaccharide in plants is starch, and starch is a mixture of 2 homopolysaccharides of glucose. Okay, those 2 are called amylose and amylopectin. Okay. Amylose is not branched. It's a linear chain of alpha 1 to 4 linked residues. So amylose is, fairly similar to glucose, at least the linear part of glycogen, being alpha 1 to 4. Okay. Amylopectin, on the other hand, is branched like glycogen. Okay, it's alpha 1 to 4 with branches of alpha 1 to 6. So in a sense, amylopectin is really the same thing as glycogen, except now these branch points occur only at every 24 to 30 residues. Okay. The molecular weight of amylopectin is up to 200 million grams per mole. Okay, so this is a very, very big polymer. Okay. And what starch looks like when these are put together, the amylopectin and the amylose are shown here. So in black would be what we would refer to as the amylopectin part, right, in black, because you have these branch points with the red dot shown. Okay. Those are intertwined with the blue here is amylose. Okay, and remember amylose have no branch points. They're just alpha 1 to 4 linked glucose. But they can kind of feed in and intertwine with the amylopectin. Okay, the non-reducing ends are shown on this side. The reducing ends would be on the right side. Okay, and remember the reducing ends are those anomeric carbons that don't have a glycosidic bond. Okay, and there are many fewer reducing ends than there are non-reducing ends. Okay, so if we look at our linkages in starch and glycogen, okay, right, this would be our glycogen. Okay, this could also be amylopectin. Honestly, what we're seeing, the alpha 1 to 4 linkages, and then also the alpha 1 to 6 branch linkages. Okay. Glycogen, if we draw this out using just the hexagons for the sugars, glycogen would look something like this because there's branch points much more frequently. Amylopectin would look a little bit more spread out because the branch points are more spread out. Okay, and then finally amylose is just a continuous chain of glucose, but it can sort of form kind of bend around itself, okay, and that's one of the reasons it's good at kind of intertwining with amylopectin. Okay. So the metabolism of glycogen and starch is again somewhat similar because they are very similar structures. They both have one reducing end, but many non-reducing ends. So what does that mean? Well, it means if you're going to design a system to metabolize these polysaccharides, you're going to want something that will process them at the non-reducing ends because there are more of them. Okay, and so if on this picture here, our non-reducing end is shown here in the red. Okay, the branch point is shown in green, and the this would be at the very end of these, not shown here on this slide, would be the reducing end. Okay, so there are many more of these reds that are reducing than the non-reducing. Okay, and that's where the enzymes are going to act on those. Okay, let's talk about some other polysaccharides. Okay, another very important polysaccharide shown on the title slide is cellulose. Cellulose is a branched homopolysaccharide of glucose. So again, this is the third polysaccharide we're talking about, and it's only using glucose. Cellulose is different than what we've seen before because it is a beta 1 to 4 linked chain. Okay, so instead of the alpha anomer, glucose is in the beta, and this is where we get this kind of funny picture of, remember the beta anomer would have an OH group coming up on carbon 1. Carbon 4 has the OH group pointing down, so when we do this glycosidic bond, we show one going up here and one going down, so it kind of looks like, I don't know, like an N almost here. Okay, so this is a beta 1 to 4 linkage. Because of this, the way this structure is, it forms additional hydrogen bonds between the adjacent monomers for one, and then also between different chains. Okay. And this structure becomes very, very tough and very water soluble, and this beta 1 to 4 linkage really makes it more of a structural polysaccharide, a structural molecule, because it can be very strong and very rigid, as opposed to something like starch or glycogen, which is more not used for structure, but all for storage. Okay. And so this shows a little bit better of what this hydrogen bonding is, right? So we have hydrogen bonds formed between, and this picture, these pictures also are better at showing you what the actual molecular geometry is than when we showed this, okay? So this is what the actual geometry would look like. We can get hydrogen bonding between adjacent monomers. We can also get hydrogen bonding between chains now, and those hydrogen bonds make this a very rigid, very strong structural material. Okay. The metabolism of cellulose is also a little interesting. Okay, this cellulose makes a very fibrous, strong, tough structure in plants. Okay, it's very water soluble. Okay, it's difficult for enzymes to act on this. Certain fungi and bacteria in lower life forms have an enzyme called cellulase that actually allows them to use wood or other fibrous plant matter as a source of glucose. Okay? Animals don't have this cellulose enzyme, cellulase enzyme to use cellulose. Okay, so they can't use cellulose as a food source. Okay, 'cause they can't hydrolyze these beta 1 to 4 linkages. Okay. You might know that or heard that cows can do this. Okay, and the other uh, example of something that's a higher level organism that can do this are termites. And the reason that they can do this is that they have microorganisms in their gut that actually produce the enzyme cellulase. Okay, so it's not that they themselves have cellulase enzyme. They actually have microorganisms inside their guts that do this. Okay. The cellulase enzymes do hold some promise in being able to process biomass into biofuels in the future. So if you're at all interested in bioengineering or alternative energy sources, this cellulase enzymes are actually, can be pretty important. Okay. Some other polysaccharides that, I wouldn't expect you to know off the top of your head or even really recognize a picture of. One of them is chitin. Okay, this is a linear homopolysaccharide of what's known as N-acetylglucosamine. So what is that? Well, it's glucose with a acetyl group, an N-acetyl group. Okay, and that happens to be attached to carbon 2. Okay, so, and we'll see more of these acetylamine groups being used. But this one, okay, just happens to be at carbon 2, okay, and it's a beta 1 to 4 linkage between these. Okay, so just like in cellulose, this beta 1 to 4 linkage leads to an extended structure, okay, very hard, insoluble material, can't be digested, and this is what insects use as in their exoskeletons and shells. Okay, so it's a very rigid, very structural material. Right. Agar and Agarose are another example of polysaccharide. Okay, this one you probably have used in lab, or if you haven't taken biochem lab yet, you will, or even biology lab. You use agar plates, agarose gels. What that is, agarose is a mixture of two sort of a repeating disaccharide into a longer chains. And it is galactose with a beta 1 to 4 linkage with 3,6-anhydro L galactose 2-sulfate. Okay, so it's a very, very complex. Again, not going to need to know this structure for this class, but if you're interested, right, at what those plates are, those agar plates or an agarose gel, it's a polymer of this repeating disaccharide. Okay. Right, so let's now talk about something called Glycosaminoglycans. Okay. They're linear polymers of a repeating disaccharide unit. So somewhat similar to what we just saw with agar. Okay. One monomer is either N-acetylglucosamine, which we've seen, or N-acetylgalactosamine. They're negatively charged. Okay. They have either uronic acids at carbon 6, which would just be an oxidation at carbon 6, or they contain sulfate esters. Okay, so both chitin and Agar sort of can kind of fit this bill. We've seen, examples of these things in those. Okay. What these make, though, having the N-acetyl groups and the uronic acid or sulfate esters that are negatively charged, it makes these extended hydrated molecules, okay, because they want to minimize the charge repulsion between the sugars, the sugars that are adjacent to one another, so these become very extended polysaccharides. And the negative charges and polar groups will attract water. Okay. These form meshwork with fibrous proteins and form what's known as the extracellular matrix. And that's in connective tissue and involved in the lubrication of joints. So Glycosaminoglycans are involved with, physiologically, they're, sort of the extracellular matrix that's really important in connective tissues and joint spaces. Okay. So if we look at a couple of these examples, one is high allyuronate, and it has a molecular weight, a number of disaccharides per chain of about 50,000. Okay, a really big number. Okay, what this is it's glucose that's been, the A stands for an acid, right? So at carbon 6 now, instead of an alcohol group, it has an acid up there and a negative charge. And it has a beta 1 to 3 linkage with a N-acetyl glucosamine. Okay, so this is a glucose that has the N-acetyl group there, and this one happens to be at carbon 2. Okay, and then that is our disaccharide unit, and that repeats by an attachment, a beta 1 to 4 attachment. Okay, so this 2-sugar disaccharide will just then repeat itself. The other one that we'll talk about is chondroitin sulfate, or chondroitin 4 sulfate. Again, you have a glucose that has an acid group at carbon 6. And again, you have a beta 1 to 3 linkage. This time it's the second sugar in the disaccharide is a galactose that has an N-acetyl group on it, just like in hyaluronate, but it also has at carbon 4 this sulfate group attached. Okay, so another negative charge. All right, how would never expect you to be able to draw these. If you saw a picture of these on the exam, how you would know what they are, you could see that there's an acid group, right? A negative charge. There's a beta 1 to 3 linkage, which we don't see anywhere else. And there's also this N-acetyl group. Okay, so that's how you would pick those out. How you would tell the two apart, obviously this chondroitin sulfate has a sulfate group on it. Okay, these would show up on a problem where I give you, certain sugars and polysaccharide structures, and I'd have you like match them with their names. Okay, and I probably wouldn't give you both of these on a problem like that. I would probably pick one or the other. Okay, some other glycosaminoglycans that you might see: keratin sulfate and heparin. Okay, keratin sulfate has a beta 1 to 4 linkage with a galactose. Okay, so galactose isn't something that I'd expect you to really recognize. Okay, this beta 1 to 4 linkage, and then it also has a sulfate group at carbon 6, and then an N acetyl group here at carbon 2 again in a glucose. Okay, so the glucose has an N acetyl group at carbon 2, and then at carbon 6 there's a sulfate. Okay, that would be the disaccharide, and it repeats then with beta 1 to 3 linkages. Okay, so again, you're seeing a beta 1 to 3 linkage and a sulfate and an acetyl group. That should clue you in. This is a glycosaminoglycan. Okay, in this case, it's keratin sulfate. Okay, and finally, heparin. Heparin is different because there's acid groups and sulfate groups, many negative groups. Okay, that's how you would tell that one apart. Okay, all four of these I sort of expect you to kind of know, but again, if I give you something in a matching problem, I'm just going to pick one of these four. Okay, I wouldn't pick all four of these and expect you to be able to correctly name all four of them. Okay, so you don't have to go crazy with trying to memorize these. Okay, so what are they used for? That's kind of what we'll talk about a little bit now. Heparin you've probably heard of, and heparin sulfate. Heparin is a linear polymer of about 3 to 40 kilodaltons. Okay. Heparin sulfate is the heparin-like polysaccharide, but it's attached to proteins. Okay. These have the one of the highest negative charge densities of all biomolecules. Okay. And what they're used for is preventing blood clotting. Okay, by activating a protease inhibitor, which, if you're curious, it's antithrombin. Okay. Binding to various cells regulates the development and formation of blood vessels. It can also bind to viruses and bacteria to decrease their ability to reproduce and infect other cells. Okay. So if we looked at a segment of heparin that's shown here, just the molecular model view of this, what the structure would actually look like. Okay. And the protein that it's binding in here, if this is our heparin, the glycosaminoglycan, short chain of it anyways, and the protein that it's binding in, you can see that it's very blue, okay? And those blue surface in this protein are positive charges. So those positive charges are attracting the negative charge of the heparin. So as a sort of a review table from the polysaccharides that we've talked about, that's given in table 7.2. Okay, and I think this is a pretty decent review. If you just want to, before an exam, go through this. Starch, remember those, amylose and amylopectin. Those are glucose alpha 1 to 4. Okay, amylopectin is the one that has the branches at alpha 1 to 6. Okay, glycogen is really, in a sense, exactly the same as amylopectin. It's just those branches occur a little bit more frequently. Okay, cellulose, that is another polysaccharide with glucose, a homopolysaccharide made up of only of glucose, but now the linkages are beta 1 to 4. Okay, this again, because of that, it's a structural, very rigid polysaccharide. Okay, chitin is very similar, beta 1 to 4 linkages, but now instead of glucose, it's a glucose with an N-acetyl group on it. Okay. And then some of the other ones we talked about, not very important, but the glycosaminoglycans are more important. And there you have acidic forms of polysaccharides, and also the N-acetyl groups are more common. And you see the beta 1 to 3 linkages as well. Okay, the final thing we'll talk about in Chapter 7 are glycoconjugates. Also, we can refer to them as glycoproteins. Okay, a protein with a small oligosaccharide attached, or maybe you can kind of think of it as a polysaccharide as well. Okay, the exact amount, a size that you would call an oligosaccharide as opposed to a polysaccharide, I really don't know the cutoff point there. Okay. Basically, what you have is a carbohydrate that's attached to a protein via its anomeric carbon. This is really actually pretty important because about half of mammalian proteins have sugars attached to them. We can refer to them as glycoproteins, which is pretty crazy because, as far as I know, this idea of sugars attaching to proteins isn't extremely well known. I'm a protein biochemist myself, and I don't really consider sugars attached to proteins very often. Okay, but half of mammalian proteins have them, so it's obviously pretty important. Okay. One of the ways that these sugars that are attached to proteins play a role, the importance that they play, is in protein-protein recognition. Right. And unlike mammalian biochemistry, bacteria, only some bacteria have these glycosylated proteins, or proteins that have sugars attached to them. Viral proteins, however, are heavily glycosylated, and that helps them to evade the immune system of their host. Okay, so how are these connected? Well, there are two primary ways in which a sugar can be attached to a protein. Okay. Both of the times it's going to occur at the anomeric carbon. So in this case, this is our sugar. It's galactose with an N-acetyl group on it. Okay. This is the anomeric carbon. Okay. In this case, it's attaching via an oxygen group on an amino acid side chain, which shown here is a serine. Okay, so this is what we refer to as an O-linked either glycan or sugar. Okay, so these O-linked sugars are either going to be at a serine or at threonines. Okay, and so these are some examples, right? If you use the little code here down in the key, you can see, the different types of sugar patterns that can be attached. Okay, you're not going to need to memorize any of those patterns. What I do hope you remember is that the O-linked means it's attaching to an OH group on an amino acid side chain, which would make it a serine or a threonine. The other way that it can attach is what's termed as N-linked. So here we have again, this is a glucose with an N-acetyl group. Okay, it's attached at its anomeric carbon, but now it's attached to a nitrogen group. And that nitrogen group is going to be from the side chain of asparagine amino acid. Okay, so N-linked attached to asparagine. And you can think of N as being nitrogen, or you can also think of N as being the one-letter abbreviation for asparagine. Okay. So a term proteoglycans. Okay, and these are, we'll see a couple example of these. They're sulfated glycosaminoglycans, that's what we've talked about before, that are attached to large rod-shaped proteins in cell membranes. So we have these glycosaminoglycans attaching themselves to a protein that's in a cell membrane. And there's a couple different types. Not going to need to know the difference between these, but syndicins, protein that have a single transmembrane domain, and glycogens, proteins that are anchored to a lipid membrane. Okay, they interact with a variety of receptors from neighboring cells, and thus they can regulate things like cell growth. Okay, so here's an example: syndicin and glycogen. Don't need to remember the differences between those. But basically you have proteins that are in the membrane, either, sort of an integral protein that itself sticks in, or it can be anchored to a lipid that's in the membrane. And then you have this chain, this inositol chain that anchors the protein down. What makes these, why we're considering them in this chapter, is that they have these sugar groups attached to them as well. And so here, shown in green, we have heparin sulfate attached, okay, and chondroitin sulfate here in orange. Okay, and those are on the outside of the cell. Okay. So different glucosaminoglycans are linked to the core protein in a proteoglycan, and the linkage from the anomeric carbon of xylose to serine to a serine hydroxyl. Okay, our tissues have many different core proteins that do this. You can have aggregates of these, so Heparan and what's known as Agar form huge, on the order of hundreds of million molecular weight aggregates. Okay, those, if you remember, they're negatively charged and very polar. These sugars, they can hold lots of water, estimated about 1,000 times its own weight. Okay, so all of that water being held there in those molecules on the outside of these cells provides lubrication. Okay, this is a very low friction material. Okay, the surfaces of your joints are covered with this cartilage. And what that does is it reduces friction, and it also helps with load balancing. So what does this look like? Well, you have this hyaluranin, shown here is just kind of a cartoon, right? But that's this core red structure here. And that is this 50,000 repeating disaccharides are making that up. Okay, so when we talked about this polysaccharide, and it was shown as 50,000 in the chain, that's kind of what we're talking about. So that is the core, and then on top of, attached to the core, I should say, are this aggregate protein, which is shown here also in red. So that's kind of probably a little bit confusing, right? This here is a polysaccharide. These are protein, rod-shaped proteins attached to it. Okay. Attached to the aggregate protein are these blue and red sort of, they look like little feelers or something, these sticks. Those are other polysaccharides, the chondroitin sulfate and the keratin sulfate. Okay, and again, those are, very polar, very negatively charged. Okay, that's where, so if we look at the big picture here, so if you have this on the outside of your cell, what's going to happen is all those negative charges and the polar aspect of the chondroitin sulfate and the keratin sulfate, that's going to attract a lot of water in those. Okay, and if you have these in that joint space with a lot of water around them, you're going to have a very low friction material that can slide back and forth and actually help cushion as well. Okay, and this kind of makes up part of the extracellular matrix. Okay. All right. The extracellular matrix as well contains some other things listed here. Not going to need to know about them in this class specifically. If you're interested, though, they're things like collagen and elastin, as well as the proteoglycan aggregates that we just talked about. Okay. This extracellular matrix is a barrier for tumor cells seeking to invade new tissues. in fact, some tumor cells can secrete a enzyme called heparanase that degrades this extracellular matrix. Okay. In this extracellular matrix, you can also have interactions with other cells. Okay. And probably the easiest way to show this is with a figure, right? And we've talked about, some of this should look familiar, right? We've talked about integrins. They're receptors that stick on the outside of a cell membrane. They can attach to things on the outside of the cell, the extracellular matrix, that triggers them to have some sort of an effect on the inside of the cell. And one of the things they can attach to in this extracellular matrix, fibronectin is shown here, but that's also associated with the proteoglycan that we were talking about, that has a hyaluronate backbone with the aggrecan and the keratin sulfate and chondroitin sulfate, right? So that this is all making up, and collagen, we've talked about collagen before and protein structure with the triple helix. Okay, that's all of this is kind of combining to form the extracellular matrix. Okay, so this really kind of shows, a lot of what we've talked about already this quarter. Okay, we don't get in a whole lot of detail about what this is actually doing, but you can at least see some examples of some of the structures that we've seen so far. Okay. One of the reasons we don't talk a lot about this is because of how complex it is. But you can see these examples of some of these molecules and structures that we've learned throughout this semester. Okay, so just in summary of chapter 7, what's really important are the structures of the monosaccharides, the really important ones, glucose, ribose, and fructose. And then we looked at some disaccharides, okay, and that glycosidic bond, how you can have anomeric carbons, okay, that's important. Some polysaccharides, the important polysaccharides we mentioned today, those are the most real pressing important parts of this chapter. Okay. The next lecture will be covering nucleic acids. That's from chapter 8. And really the 3 components of a nucleic acid. One is the base. Okay, the bases you probably know from biology. You have pyrimidines and purines, right? The A, G, C, T, those letters are the bases. Okay. Other part of the nucleic acid that's important is you have a 5-carbon sugar, and then you have phosphate groups as well. So we'll talk about the basic structure of nucleic acids, and then the actual discovery of the double helix structure in DNA.